Our next speaker is Marcella Birtele from USC.
Okay. Okay, great. Thank you. Hi, everyone. It's a pleasure for me today to talk a little bit about my postdoctoral project that I'm conducting here at USC in the Quadrato Lab. So my focus is around neurodevelopmental disorders. So these disorders have high clinical heterogeneity, and this stems mainly from the fact that they're linked with polygenic mutations, and also due to the fact that each individual genetic background really plays a role in the clinical phenotype of these patients. And so when we study these disorders, we know that high-order cognitive functions are associated with specific areas within the cortex. And in the human, we have this really mapped out in the human brain. However, when we want to study these disorders in the lab, we mainly use rodent models. But what we need to consider is that we have differences in the cytoarchitectural structure of the cortex. So if we see here, there's a schematic of the cortex of rodents and human, and we see there are specific layers within the human cortex, such as the subventricular zone that is populated by outer radial glia that is not present in rodent models. And so we find these specific cell populations that are not present in animal models. However, when we want to work with human tissue, especially human fetal tissue, we have very limited access to these samples. And the samples that we can get are really just a snapshot of a specific stage, and they really don't show and allow us to see all the developmental biology that is occurring at these early stages of the brain development. And so we heard a little bit about this regarding brain organoids, but really there is a need for a human-specific model system. And so in the lab, what we can do is to derive stem cells, so induced pluripotent stem cells from patients and also from healthy controls. And what we can do then is to differentiate these stem cells towards different fates. So what I've been working on has been differentiating these cells towards brain, so cortical organoids. So this three-dimensional structure in vitro. And I've been leveraging these organoid models to study specifically SYNGAP1. So SYNGAP1 mutations have been linked with intellectual disabilities, developmental delay, autism, and many other clinical symptoms, including also gastrointestinal disorders. And I'll touch upon these last symptoms in the last part of my talk. But mainly my project has been focusing on leveraging cortical organoids to model this disorder. And so when I started this project as a postdoc in the lab, what was known about SYNGAP is that it is a protein that is really important for the postsynaptic density of excitatory neurons. However, all the knowledge available was deriving from rodent models. So being an electrophysiologist, what I wanted to do was really to understand the role of SYNGAP within a more human physiological models, such as organoids. So starting this project, I wanted to look at the expression of SYNGAP at the different developmental stages of the brain. So here you see a single cell sequencing data set that was publicly available, and I leveraged this data set to look at SYNGAP expression. And in the red here, you see the expression of SYNGAP and the enrichment of SYNGAP1 in different cell types. So what we could see was that SYNGAP was expressed in the neuronal component, as was previously shown to have a role in this cell population. But what was really surprising to us was the expression of SYNGAP in radial glia cells. So this was something that was unknown before, and it really changed the direction of the project. Because from here we started to wonder, okay, what is SYNGAP doing in these cell types that are the progenitor of the cortex? So we used cortical organoids. You see here stainings for 2-month-old organoids where we have SOX2 positive cells in green, labeling progenitor cells. And they're really organizing in these ventricle-like structures. So we wanted to confirm what we saw at the single cell level. So we looked for the SYNGAP protein. And here in red, you see the expression of SYNGAP within that SOX2 positive compartment of progenitors. And particularly in that area, we see SYNGAP1 being expressed in the apical domain of the ventricle. And we wanted to confirm this even further, so we collaborated with the COBA lab at USC, and we performed the mass spectrometry at day 7 organoids where we have a homogeneous population of progenitors. And indeed, we found the peptides for SYNGAP, but not only that, we also confirmed our staining results where we saw among the SYNGAP interactors also TJP1, this tight junction protein that we see colocalizing in white here with SYNGAP1 at the ventricle of these organoids. So next, we started to wonder what's the function of SYNGAP in radial glia cells. So we performed bulk RNA sequencing from 2 different types of organoids. We derived organoids from a patient carrying a point mutation, this Q503X mutation, that give rise for haploinsufficiency of the protein. And we compared it to organoids generated from the same line, but we CRISPR corrected the mutation. So you see here the differentially expressed terms that mainly give us an idea of SYNGAP being associated with the cell locomotion and migration. And when looking at the interactors of SYNGAP, we found that most of these proteins were related to cytoskeleton. And so this really suggested us that SYNGAP at these early stages might have a role in cytoskeletal remodeling of radial glia cells. And cytoskeletal remodeling in radial glia cells in the context of cortical development is really important. As we see here in a schematic, the radial glia cells are these cells laying around the ventricle, close to the lumen, and they extend 2 different cytoskeleton compartments, one apical towards the ventricle, and here this is a really important compartment for the maintenance of the ventricle. And on the other side, this radial glia really extend a long cytoskeletal protrusion towards the basal side of the cortex. And this is really important for the migration of neurons. So we started to look at early stages of radial glia cells organization and development using a 2.5D assay in collaboration with Randy Ashton. And here you see the formation of single ventricle, also called the single rosettes. And we compared the patient with the corrected progenitors. And what we could find, and you see here from the staining, is that with the patient progenitors, we couldn't see the formation of this single ventricle or single rosettes, that instead we really see very well organized and formed within the corrected lines. So really suggesting that the haploinsufficiency for SYNGAP is affecting radial glia scaffolding. And we also added additional lines where we started from a control line and we inserted the patient mutation. As I mentioned at the beginning, for many of these disorders, the genetic background is really plays a role in the clinical phenotype. So to investigate that, we inserted the patient mutation in a control background. However, what we saw was that progenitors in this condition were still not able to generate single ventricles, suggesting that in this case, the genetic background doesn't play a role in the phenotype. And finally, we also targeted a specific region of the protein, the RASGAP domain, and we rendered this domain non-functional. So you see here in the quantification that most of the progenitors were not able to form these single rosettes, suggesting that this specific domain of the protein is the one that is involved with the cytoskeletal remodeling of radial glia cells. And so next, we wanted to look at the later time point when in culture we have not only progenitors, but also we have neurons. So we waited for 2 months or so, and here you can see a staining of an organoid from a control and from the patient. And we stained for different markers of neuronal population, such as CTIP2, SAPB2. And when we look at the patient organoids, what we could see was the presence of these neurons within the ventricle, so within the progenitor zone, something that we haven't seen, never seen in the controls. And when looking even further at the radial glia cells laying around the ventricles, we observed their differentiation or their division mode. And what we could find was that SYNGAP patients had an increase in differentiative divisions, suggesting that this SYNGAP haploinsufficiency impairs the developmental trajectory of radial glia cells. And to really dissect this population of cells, we performed single-cell RNA sequencing at 2 months. So here you see patient organoids that we compared with corrected derived organoids. And it was really striking to see that focusing on apical radial glia, these cells in the patient have downregulated terms for mitosis and instead an upregulation of terms for neuronal differentiation, again indicating that there is an impairment in the developmental trajectory of these cells. And finally, we performed also calcium imaging and single-cell RNA sequencing at 4 months. I'm not showing here the single cell, but with the calcium imaging, we could see in the patient and also in the control line with the patient mutation that there was an increase in activity, suggesting that decrease in SYNGAP1 accelerates neuronal maturation. And with this, I hope I've shown you how with organoids, cortical organoids, we have identified an unknown function for SYNGAP in regulating human cortical neurogenesis. And this reframes our understanding of commonly known synaptic proteins and how these proteins might play a role at early stages of development. But also the fact that these proteins and genes might have a role in different cell types other than the commonly investigated cortical neurons. And this led to this second project that we're currently investigating in the lab. And these projects mainly start from meeting the patients and also with the caregivers, we really had the feedback that one of the most impairment from the patient is related to gastrointestinal dysfunction. And so, similar to the cortical project, I started to look at the expression of SYNGAP and all the top ASD-associated genes in the gut tissue. And so here we have, I started from an available nuclei sequencing of gut adult tissue, including enteric neurons and other tissues. And I plot here the top 102 ASD-associated genes included targeted SYNGAP1. And very interestingly, I found expression here in panel C of SYNGAP1 in a specific subtype of enteric neurons. So this is a current project in the lab. We started to generate enteric neurons, and already at day 30 we have mature 2D neurons. And we performed a preliminary screening where we generated enteric neurons from patient, SYNGAP patient, and control. And from qPCR, we can see that already at day 12, they start expressing increased levels of maturation markers. And this was also confirmed by calcium imaging analysis. But I won't tell you more about this story, and I would invite you to visit poster number 163 from Michelle, who is an amazing CIRM Bridges, and she's really pushing this project in trying to generate an enteric nervous system organoid model to dissect even more this first phenotype that we observed. And with this, I want to really thank my amazing mentor, Giorgia Quadrato, all the people in the lab, all the funding agency, and my mentors. And thank you for listening.
Hi, really interesting work. So later in development, radioglia start making astrocytes. Do you see expression of SYNGAP1 in astrocytes and any differences there?
Yeah, so our studies were mostly done between 2 and 4 months, where we still don't have astrocytes in cultures. So we see astrocytes from 5 to 6 months. So we haven't looked at that, but Marcelo Cobba at USC is actually focusing on the role of SYNGAP in astrocytes. So yeah, I don't know the details on that, but yeah, it seems to play a role also in the astrocytes.
All right, thank you.
Yeah. Thank you.